Hello, we want to run through the Plant Reproduction Lab or Plant Reproductive Structures Lab. So right here's Plant Reproduction Lab. We're going to go over here and click on Run, open up the lab. While that's opening up, let me remind you that you will see the short answer questions that are open in this lab. You don't have to do those. I have to have those short answer questions open so that I can see your photos in the Your Media section. So you're not actually doing the short answer questions. Okay, the other thing is I want you to mainly follow the directions on your lab handout rather than the directions in the procedures on Late Nite Lab. Okay, we've got our lab open. We'll get our lab manual up here in just a second. Here we go. So I'm going to open that up, and there's the background. You can kind of scan down through that background if you like. There's some information in there, but it's kind of hard to follow without diagrams. The way that it's presented is a little bit hard to follow. Now, here's what we want to look at. We want to go over here to Procedures, Getting Started, and in this lab you're going to use the Media Player, which you've used in other labs. And so this first section right here just goes through a description under Getting Started, goes through a description how to use the Media Player, how to use the draw button, and how to save your images. Okay, so the first thing that we want to do is we want to go down here to the section on the male pine cone. So we're going to get our microscope, get that, materials. We want to find there's a female pine cone, male pine cone, put that on right there. And let's go over here where it says coarse focus. I'm going to roll that down, bring it into focus. Okay, now then, I'm going to go over here and open up Media Player. And here it shows a longitudinal view of the male pine cone. What we're looking at over here is a cross-sectional view of this male pine cone. Okay, I ask you to take a snapshot and then identify two parts, cone axis and the microsporangium. So let's go in a little closer. I'm going to go down here to this plus sign, bring this in a little bit closer, a little closer, a little closer. Now then, I'm going to just grab the whole screen and slide it over again. Now then, got everything where I can see it. And I'm going to take a snapshot of this. This button on the microscope right here, it says snapshot. There we go, got the snapshot. Let me move this out of the way, and I'm going to entitle this, well, maybe I am, Male Pine Cone. Okay, I'm going to save that. There we go, snapshot is saved. So now then I want to go down here to where it says Your Media. Okay, well, it says I didn't save it. Okay, so let's close that. Let's try that again. It doesn't want to work, so I'm going to completely close that out. I'll reopen it. Now let's try it. Now it's in there. Okay, so I had to close the Media Player and then reopen it to see my photo. Okay, so I'm going to use the drawing tool, and I'm going to use red. And right here, all of these little structures that you see, those are the microsporangia. So I'm just going to circle one of these right here. Microsporangia, go out here, and I'm going to label that. Okay, so, and let's see, I'm going to label that in red because there's so much black in that photo. So, there we go, let's try that. Okay, so, microsporangia. Okay, sporangium, since there's actually just one of them. Sporangium. Now then, I've got a bit of a mistake there. I ran out of room. Should have allowed myself more room. Okay, let's go back to drawing again, and let's mark right here this structure, and let's label that as the, let's try blue this time. The red didn't seem to show up all that well. I don't think blue is going to either. Okay, let's label that central axis. No, blue doesn't show up very well either. But the central core right here, that whole thing, that is a central axis, and then these structures are growing off of that. So each of those light colored chambers is a microsporangium, where the process of meiosis occurs and produces what's called microspores. The microsporangia are essentially the plant equivalent of testes. Microspores are the plant equivalent of the sperm cells. Okay, so let's go to the next part on this, and let's go back here to media, and let's reset the position in the lab. We'll take our slide off, put it in the trash can perhaps. Well, it went somewhere. Okay, so let's go to male pine cone. That was male pine cone. Let's go to pollen. So female pine cone, male pine cone, pine pollen. Put that on the scope. Go to coarse focus adjustment, roll that down, bring it into focus. Now then, on your sheet, it tells you that we need to run this up to about 4x or 8x. Okay, so let's go in closer, 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 and slide it over so that we can see this. There's 2x. Here's 4x. That's pretty good. I'm going to slide this over. Here's my x-axis adjustment. I'm going to slide that over right here. I want to see if I can find a good example of this winged pollen, one that looks kind of like Mickey Mouse, and right there we go. So I want to get a snapshot of that, and I'm going to label this as pine pollen. Move this out of the way. Okay. And okay. Now, once again, I probably need to close this, reopen it, so that I can find in your media. There's my pollen shots, and I want to label this, and I'm going to label this right here with the drawing tool. Let's go right here with black, and so here's the right there and right there. Those structures are the wings, and I need to type wings out there. Now then, right here, see, there's allow some space where I can put in the label. The dark spot inside, that's the nucleus. So I would use my text tool, and we'll go in and label that as the wings, that as the nucleus. Okay, so let's go back to Media Lab. Let's put this back in its original position, reset the zoom. I'll move this up out of the way, take the microscope slide, whoa, not the whole microscope. Take the microscope slide, put that in the trash. Well, I'm not going in the trash. Oh well. Okay, now the next one we want to look at is female cone, and we'll do the same thing here. We're going to bring it down, place it on the scope, bring that into focus, and we want to take a snapshot at 1x. So go in a little bit closer, 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 closer. We'll move this over, and also let's go down here to our female pine cone diagram. Okay, so I'm going to take a snapshot at 1x. So female pine cone 1x. Okay, I'll save that. Okay, now I want to go up to, there's 2x, and let's go up again to 4x. And what we can't see in this one right here is the micropile over here, x-axis. Let's slide that over, and we can see the micropile in there. Okay, so I'll get a snapshot of that. So female pine cone, that's 4x. Save that. Save. Okay, so I want to open those up. Well, that's already open, so for them to enter, I got to close that, reopen it. Your media, here we go. Okay, let's open this one. Now then, use my labeling tool, and right here, these structures like this, right here, this, right here, here's another one over here like this. These are the, I'll label these structures as, use texting tool and label that as the megasporophyll. Phyll refers to leaf, and these structures are modified leaves that become the bracts or the scales on the cone. Then the seed develops here on the surface of that scale. Okay, so I want to save that, and I'll need to, I should have saved the others a while ago, I didn't. So I need to save that, I need to give it a title, and of course you need to give it an actual title there. We'll go that route. Then let's go over here to your media. Let's go down to the one that is a little bit closer. Now then, we want to mark these. This is the megaspore right here, which is the equivalent of an egg cell. This would be the integuments right out here and right around here that will become the seed coat, and the opening right there is the micropile. Okay, so I would use my drawing tool, I would draw lines to those, use the labeling tool, label that, and then I would save it. Okay, let me get that out of the way. Now then, the other one that we want to do is we want to take this off, that out of there, and the other one that we want to look at is the wheat seed. We're going to put that on there, bring that into focus, and let me just show you the parts on here. Okay, right here around the very outside we have the seed coat. Right there's a good view of it, seed coat. All of this tissue in here is the endosperm. Now then, let's go in a little bit closer, or you'll need to go in a little closer. This is going to be the cotyledon right here. This structure, the pointy end, is the epicotyl. The blunt end is the radicle. So take photos of that, mark it, and then save it, and I'll take a look at it and see how well you did. Thank you. Hope you do well with this lab.